Dear students, welcome to the online classes of biotechnology. And today I will explain you about cloning vectors. As you know, last lecture I have explained about some plasmid vectors like PBR322, PUC vectors. In continuation of these vectors, today I will explain you about Cosmid vectors, bacteriophages vector, and finally I will discuss about some viral based vectors, particularly for insects and mammalians. So first I would like to introduce you about the Cosmid. Actually, Cosmid is the hybrid of here you can see it is the hybrid of fast DNA and plasmid. So it contains a part of lambda DNA or fast DNA containing the cos site and some part of the plasmid DNA. Here you can see the plasmid part contains BAM-H1 site, which is for the cloning purpose, ampicillin resistant site, and origin of replication. Similarly, the fast lambda contains the cos site. Why this cos site is so important? Because it is actually responsible for the packaging of viral DNA in the capsid of the viral particle. So we use this cos site here. So in figure B, you can see how this PJB8, actually this is the name of the Cosmid vector. So actually this can be linearized or digested with the BAM-H1 site. Here you can see the many BAM-H1 digested PJB8 vectors or Cosmid vector, and then gene of interest, which is also digested with the BAM-H1. They can be ligated in a manner such that first it will contain the cos site, then ampicillin resistant site, then gene of interest, and then again cos site, then ampicillin resistant site, and gene of interest. So these linearized form are called catenane, and these catenane then finally packaged in the bacteriophages by the in vitro packaging. So they are easily packaged into the capsid of the bacteriophages because they contain the cos site. And once they are packaged in the fast particle, they are transpected or infected into the E. coli. So these fast particles are unable to produce any plaque in E. coli because they lack all the plaque forming genes, and Cosmid with foreign DNA is cyclized in the bacteria cell and can replicate as a plasmid. So once the bacteria replicate, so these plasmids can also be replicated. So here you can see how the Cosmid is quite important for the cloning of a long fragment of DNA up to 45 kb. So it is very important for Cosmid. It can clone up to 45 kilobase pair of DNA. So that's the beauty of Cosmid. So another type of cloning vector is bacteriophage. We can also use bacteriophage as a cloning vector. Why? Because of its transduction capacity. So as you know, what is transduction? Actually, in transduction, what happened? The phage can introduce its DNA into host cell. Here you can see the host cell is a bacterial cell, and the DNA of phage particle is shown in red color. So once it inserted into the host cell, it can replicate, and the virus capsid and virus assembly takes place inside the host, and the packaging of the viral DNA takes place in the capsid of the virus. But what happened? Occasionally, this capsid is packaged with the bacterial DNA. It happened in some cases from 1% to 5% cases. And once this host cell lysis takes place, this phage particle containing the DNA from the previous bacteria can infect a new bacteria. Here you can see this bacteriophage, which is containing the DNA from the previous bacteria. It can inject the bacterial DNA into the new host cell, and the integration of this DNA which is from the previous bacteria, it can integrate into the new host genome. So here you can see this is the stable type of gene transfer takes place because of the transduction capacity of the bacteriophage. So we can use this generalized transduction capability of this bacteriophage as a cloning vector. Now I will discuss about the cloning vector for insects. As you know, the Drosophila is most vastly used organism in the insect genetics. So the P element is actually used as a cloning vector for the Drosophila. What is P element actually? It is a transposition or jumping gene. So as you know, what are the transpositions, and these are the actually found in the all organism, and they are the short piece of DNA, and usually they are less than 10 kb in length. They can move from one position to another position in the chromosome of the cell. So the P element, which are one of the several type of transposition in Drosophila, are 2.99 kb in length and contain 3 genes flanked by short inverted repeats. So in next figure you can see the position of these transpositions or P elements in Drosophila. Here you can see the 3 genes for the transpositions genes, and they are flanked by the inverted repeats. Here you can see this is 5' inverted repeat, and this is 3' inverted repeat. These are called terminal repeats. In figure B you can see how this P element can be transposition into the host chromosome. So here if you will introduce this P element containing gene of interest, and if you will transpect into the embryo of Drosophila, it can be jump from this position to the chromosomal region of the host DNA or in the Drosophila DNA. And in figure C you can see the structure of a P element cloning vector. Actually the P element cloning vector contain 2 type of elements. One is P element containing the inverted repeats having the restriction site for the cloning purpose, and another element P element is also there but it lacks the inverted repeats, so it is called a wing-clipped element. So why we put this wing-clipped element in this vector? It's because when you disturb this gene by digestion and you introduce a foreign DNA in this region, so it will lose its capability to jump from one position to another position, or it will forget where it has to introduce the gene of interest in the host genome. So this wing-clipped element actually guide this P element where the gene of interest should be integrated. So we keep both elements with inverted repeats as well as without inverted repeats. It's clear from this picture here. I have shown you an example of integration of rosy gene. Actually rosy gene is a gene responsible for the synthesis of some important enzyme like xanthine dehydrogenase, and the mutant which lack this xanthine dehydrogenase enzyme, they have the brown colored eye. So these mutant which lack the XDH gene, they will have the brown color eye. So when we introduce this rosy gene, here you can see this rosy gene is cloned in the P element here, and this is P plasmid vector, P vector, it is called P vector. So you have introduced the rosy gene in the P vector, and you can micro inject this cloned rosy genes into the embryo of the Drosophila. So what happened inside the embryo? Here you can see the Drosophila DNA. This P element actually contain the rosy gene, and the P element has capability to jump from one place to another place. So what happened? Due to the homologous recombination, or it's a jumping or transposition capability, this rosy gene is introduced or integrated into the host DNA. So here you can see this P element is introduced into the host DNA, and this contain the rosy gene. So one chromosome is replaced by actually the one chromosome is introduced by the rosy gene now, and each and every cell now will produce actually the rosy gene in the progeny which are developed from this embryo. Now all the descendants of this embryo will contain the normal eye color, that is red eye color. So here you can see how we can use the P element vector to convert the brown colored Drosophila into the wild type red colored Drosophila. So you can convert the mutant into the wild type here using the P vector. Apart from that, there are some viruses. You can also use them as a cloning vector in the mammalian system. And first is SV40 virus, and this virus is capable of infecting several mammalian species and following lactic cycle as well as lysogenic cycle. The genome of SV40 is 5.2 kb. So here you can see the genome of this SV40, and the beauty of this genome is it is a circularized one, and you can have many restriction sites also, so you can clone your gene of interest in any of these sites. So here I have shown you the example of how you can introduce the rabbit beta globin gene in the SV40. So it is introduced in the BAM-H1 site here. So once it is cloned, you can transfect into the mammalian cell, and the rabbit beta globin gene or protein can be expressed into the mammalian host. Apart from SV40, there are some other mammalian virus based vector like adenovirus, which is enable DNA fragments up to 8 kb to be cloned. So it can clone up to 8 kb of DNA, which is longer than SV40 vector. Now the adenovirus are more difficult to handle because of their genome size is very big. Another viral based vector in mammalian system is papillomavirus, which also have a relatively high capacity for insert DNA, like BPV, which is bovine papillomavirus, which causes wart in cattle. It's particularly attractive because it has an unusual infection cycle in mouse cells. So taking the form of the multi-copy of plasmid with about 100 molecule present per cell. It does not cause any disease in the mouse cell. So mouse cell mouse cell can be survived once you transfect it with the papillomavirus, and you can introduce any new particle in the papilloma, any foreign DNA into papillomavirus, and you can transfect into the mouse cell. So it can act as a subtle vector consisting BPV and E. coli sequence and capable of replication of in both mouse as well in bacterial cells. So papilloma viruses are actually the vectors which can be used as a subtle vector between E. coli bacterial cell as well as in the mammalian cell. Similarly, the retrovirus is another example of the vector or virus based vector which are most commonly used vector for the gene therapy. Actually these retrovirus these days are used in the gene therapy. I will explain about gene therapy in some other lectures. So that's all about Cosmid and other plasmid vectors. In next lecture I will discuss about yeast artificial chromosome, that is YAC, and I will also discuss about DI plasmid. Till then, bye bye, take care.